Good morning, all of you. In the last class, we has been discussed regarding Ascomycotina and as well as Phycomycetes, who has been discussed. In this session, today we are going to discuss regarding Basidiomycetes. Clear? Basidiomycetes. Generally, it is also called as club fungi. It is also called as club fungi, and it is also known as mushrooms. It is also known as mushrooms, and it is also called as puffballs. Puff, puffballs. And some scientists have said that it is also called as bracket fungi. Bracket fungi. Clear? The today topic is Basidiomycetes. Generally, it is called as club fungi. It is well known as mushrooms, puffballs, as well as bracket fungi. The occurrence is generally it grow on soil, but terrestrial in nature. And some of the fungi are parasites, and some are saprophytic in nature. Some are parasite as well as some are saprophytic in nature. If you observe mycelium, mycelium is branched. Mycelium is branched. Septa is present. Septa. If septa is present, the nucleus is uninucleated. Uninucleated. Clear? Next. So this point is very important here. Between two septa, one large pore is present, or on septa, one large pore is present. On septa, large pore is present. Large pore is present. Such a septa is called as Dollifort septa. Dollifort septa. Clear? Here, so this Dollifort septa, the pore is covered by small membrane called one small membrane. So this membrane is called as perintosome. This membrane, the membrane which is covered on pore, such is called as what? Perintosome. Now it is looked like as dome shape. It is looked like as dome shape. So that's why the septa is called as due to the shape. Which shape? Dome shape. So that's why the septa is called as Dollifort septa. It is called as Dollifort septa. Generally, mycelium is mostly unicellular, uninucleate. But sometimes during sexual reproduction, binucleated condition. During the sexual reproduction, you can notice that most of the mycelium cells are binucleated condition. Let's see how binucleated condition is formed. Clear? Because only one reason, that is clump, C-L-U-M-P, clump clump connection. Due to the clump connection, binucleated mycelium is formed. What is clump collection? Clump connection. Here is a tubular structure or tubular connection. There is a tubular connection to one mycelium to one cell, to another cell. Through this tubular connection, one mycelium, it means one hyphae cell containing nucleus is transferred to another cell. Like one cell containing nucleus is transferred to another cell, and again this set of cell containing one nucleus is migrated or transferred to another cell. So due to that, at the last, all cells are containing two nucleus conditions are formed. Clear? Due to that, binucleate condition formed. Due to that, binucleated condition is formed. Let's see, very simple here. This is normal one mycelium at the beginning. 1, 2, 3, 4, 5 cells are there. Clump connection here from here to here, and again here to here, like. Due to that, what happened? Now this nucleus came here. Now it will become as two nucleus, and this nucleus came here, or this nucleus is come this side, like binucleated condition is formed. Here, next character is reproduction. Here, reproduction is asexual reproduction or asexual spores are not noticed. Clear? In Bacillium mycotina, asexual reproduction or asexual spores are not noticed. Here, next one is sexual reproduction. Sexual reproduction is present, but here sexual organs are not noticed. Sexual organs are not noticed. Sexual organs are not noticed. But here what happened? Generally, somatic cells. Here, fusion of somatic cell or fusion of vegetative. Here, somatic cells or vegetative cells are going to fuse. Generally, such is called as somatogamy. It is called as somatogamy. Simple. What is somatogamy? The fusion of two somatic cells or two vegetative cells. These fusion of two somatic cells or vegetative cells are genetically different. Or different stage. Cells are going to fuse. It means, for example, one cell is, this is one mycelium, here positive strain, this is one more cell, negative strain. So these two somatic cells or vegetative cells are genetically dissimilar. Plus strain and one is minus strain cell. Simple, this fusion of two somatic cells is called as what? Somatogamy. In this case, true sexual organs are not noticed. Next, true asexual spores also be not noticed during the asexual reproduction. Clear? But here, Basidium equina, you can notice the fragmentation. Through fragmentation it can propagate. Clear? So this is regarding general characters of Basidium equitina and asexual as well as sexual reproduction of Basidium equitina. Next, sexual reproduction. Sexual reproduction. During the sexual reproduction, here two somatic cells are come together and fuse. So such is called as somatogamy. Now only has been discussed. After somatogamy, again it is going to follow. First one is plasmogamy, and as is well. Second one is karyogamy, and third one is meiosis. Problem is meiosis. Here, true sexual organs are not noticed. Genetically dissimilar somatic cells are come together and fuse. Later, it is again following these three steps. First one is plasmogamy, second one is karyogamy, third one is meiosis division. Here, after the plasmogamy, dikaryon condition is formed. Dikaryon condition is formed. After formation of dikaryon condition, here clump connections are formed. Clump connections are formed. Due to the clump connections, complete mycelium is become as binucleated. The complete mycelium is become as binucleated or dikaryon. Dikaryon conditions are formed. Clear? Next, after the formation of dikaryon condition, now the hyphae cell is become as basidium. So it is become as basidium. Within the basidium, two nucleus are present. Within the basidium, two nucleus are present. Within the basidium, karyogamy is takes place. Now the nucleus is become as diploid. Within the basidium, again this diploid nucleus is undergoes meiotic division. It undergoes meiosis division to produce haploid spores. Such spores are called as basidiospores. Now these basidiospores are which in condition? Haploid in condition. Clear? After the formation of basidiospores, now this entire structure is look like as club-like structure. Entire structure is look like as club-like structure, and due to the it is also called as club fungi, and such a shape is called as fruiting body is basidiocarp. Basidiocarp is a fruiting body of Basidiomycetes. Clear? So this is a sexual reproduction in Basidiomycetes. If you observe the difference between Ascomycotina to Basidiomycotina, simple. In Ascomycotina, what happened? First, ascus is formed. Now ascus is become as ascocarp. Ascocarp is formed. In within the ascocarp, ascospores are formed. Ascospores are formed, which are in haploid condition. Clear? Next, basidium. Here, basidium. Fruit body is basidiocarp. Fruit body is basidiocarp. After meiotic division, spores are formed, such is called as basidio spores, which are also haploid condition. Then what is the difference between Ascomycotina to Basidiomycotina? Only one thing is here. In Ascomycotina, spores are formed in within the ascocarp. So here, ascocarp, these are endospores. These are endospore type of spores. Next, in Basidium here, so this is Basidiocarp. These are Basidiospores. Basidiospores. Now spores are formed on Basidiocarp in external environment. So these spores are what? Exospores. Exospores. So remaining complete phenomena is same. So this is only major difference between sexual reproduction of Ascomycotina to Basidiomycotina. In Ascomycotina, spores are endospores. In Basidiomycotina, spores are exospores. Clear? Let's see the diagram first. Here, this is one somatic cell. Here also one somatic cell. At the beginning, these cells are septate, uninucleus condition. Now, somatogamy, again plasmogamy. Now what happened? These two cells are fused to form of like this one. So this condition is called as what? Dikaryon. Dikaryon condition is formed. After the formation of dikaryon condition, again what happens? Clump connection is formed. From here to here, one tubular structure is formed. Now from here to here, one tubular structure is formed. Due to that, what happened? Now the complete mycelium is here two nucleus, binucleated. Here also two nucleus, binucleated. Here also two nucleus, binucleated condition is formed. Now we can call as binucleated condition, and it is also called as dikaryon condition. After the formation of this condition, now Basidiocarp is formed. This is Basidium. Clear? is in Basidium. Now two nucleus are there. These two nucleus are going to fuse. Nothing but karyotami. Now we are going to write directly two nucleus. Karyotami takes place to form diploid nucleus is formed. In within the, now it is called as what? Basidiocarp. This is called as basidiocarp. In within the basidium, here again meiosis business takes place to form here outer surface uninucleated non-motile spores are formed. Uninucleated non-motile spores are formed outer surface of the basidium or basidiocarp. So these are basidiospores. These are basidiospores. Now during the favorable condition, these basidiospores are germinate and become as one mycelium. Clear? So this is actually sexual reproduction of Basidiomycetes. Some examples of Basidiomycetes. In NCERT, they given rust, smut, the other one is agaricus, nothing but mushroom. So these three examples are given in NCERT. Let's see first one, rust. Rust disease. Rust disease is mainly caused by Puccinia member. Pucci, Puccinia member. Caused by Puccinia member. So this Puccinia fungi spores or give the color, or the spores are like red color, orange color, brown color. Spots are formed on the leaf or stem or stripe of the leaf. Clear? So some of the small information regarding Puccinia. First one, Puccinia. The Puccinia it need two hosts. Puccinia member it need two hosts for completion of life cycle. For completion of life cycle. Completion of life cycle. The first host is called as wheat primary host, and secondary host is barberry. Clear? In NCERT, Basidiomycetes examples are rust, smut, agaricus is given. Actually, the rust it means brown color, red color, orange color. Spots are formed on the leaf or stem or stripe of the leaf. So these orange color spots are generally, it is not a mycelium, it is a spores of the mycelium. It is a spores of the mycelium which is give the color to the red or black or orange color. Here, first regarding Puccinia, it is a rust. Generally, it needs two hosts for completion of life cycle. The first host is wheat, it known as primary host. Secondary host is barberry. So that's why it is also called as heterodicious. Heterodicious fungi. Next, during the life cycle of Puccinia, five different types of form, five different types of sports form, five different types of sports are form. Clear? Next. So this disease is discovered by K.C. Mehta. K.C. Mehta was first to discover the disease. Let's see example of Puccinia members. Puccinia graminis, Puccinia recondita, Puccinia graminis, Puccinia striformis. Puccinia striformis. First one is Puccinia graminis, Puccinia recondita, Puccinia striformis. So these are cause the leaf. First one is stem rust. It causes stem rust. It is on leaf rust. It is on stripe of the leaf, stripe rust. So this is one of the examples of rust. Next one is smut. These are black in color. Black colored spots appear on leaf as well as stem. Species is Eustylago. Eustylago species. One example is enough. Next one is Agaricus, nothing about mushrooms. Agaricus, nothing about mushrooms. You know very well mushrooms are mostly terrestrial. They grow on soil, wet soil, moisture conditions, as well as mainly they grow in a rainy season on wood or on trunk, that we can notice. And mushroom, it is generally looked like as the mycelium is multicellular. The mycelium is multicellular. The shape is umbrella shape. Umbrella shape. Clear? Let's check. So these mushrooms, some of the mushrooms are edible. Edible. Example is Agaricus bisporus. Agaricus bisporus. Some Basidiomycotina member are edible, such as Agaricus bisporus or bisporus. Agaricus bisporus. Here it is looking as gills of fish. So that's why it is also called as gill mushroom. It is also called as gill mushroom. And some of the mushrooms are poisonous. Very poisonous. Such poisonous mushrooms are Amanita. Amanita species. Amanita. So such is very dangerous, very poisonous to the health. Which one is Amanita mushrooms? Amanita mushroom. And last example is we given puffball. Puffballs. Puffballs. Here generally puffball, it is a fruiting body of Basidiomycetes. It is a fruit body or fruiting body. So the fruiting body is large and color and beautiful in a attractive manner. Puffballs generally here it is a fruiting body. The fruiting body is large in shape and color and very attractive in manner, beautiful nature. So when we are going to touch of puffballs, it is immediately going to burst. And you can see it going to be the velocity, some speed it going to, through speed the spores are come outside from the fruiting body. Clear? So cheese one is what? Puffballs. Example is clavatum, clavatin, Deuteromycetes. Deuteromycetes, it is also called as fungi imperfecta. Fungi imperfecta. It is also called as fungi imperfecta. Why? Because Deuteromycetes members, in this case here, true sexual reproduction is not recognized till yet. There is no true sexual reproduction. They are not recognized till yet. So that's why the species are called as fungi imperfecta. They belong to Deuteromycetes. What I will write here? True sexual reproduction is not recognized yet. Or sexual reproduction is not noticed. So that's why they are called as imperfecta fungi or Deuteromycetes. Here normally these some are parasite and some are saprophytes. And majority of the members they are growing on decomposers of litter, which is also useful in mineral cycle. Which is also useful in a mineral cycle. Clear? First, Deuteromycetes. It is also called as fungi imperfecta. Perfect stage of sexual reproduction is not noticed. So that's why it is called as fungi imperfecta. Next, generally they grow like as parasites, saprophytes. Majority of the members they grow like decomposers of litter, which is useful in mineral cycle. Next, if you focus on mycelium, mycelium is branched and septate. Septa is present and uninucleated mycelium. This is regarding mycelium. Branched, septa is present, uninucleated. Next, reproduction. The reproduction I noticed only asexual reproduction through conidiophore. It is producing conidia or conidia. Clear? Next. So true sexual reproduction or sexual stages are not noticed. Now the still investigation is going on. If any one of the Deuteromycetes member, if it is shows sexual stages, then according to the, if it is shows Ascomyctina like sexual reproduction, or it is removed from Deuteromycotina and kept in Ascomycotina. If sexual stages are similar to Basidiomycotina, So these fungi are deleted from Deuteromycetes and added in Basidiomycetes. If sexual reproduction is not recognized based on mycelium as well as asexual reproduction, we given Deuteromycetes. Clear? Are you got the point or not? Very simple here. If sexual reproduction is noticed, if if it is like to Ascomycotina, then it is added in Ascomycotina. If it is reproduction method is look like as Basidiomycotina, then it is considered as Basidiomycotina and such is deleted from Deuteromycetes and added to the Basidiomycetes. Clear? Next, examples. Example is Alternaria solani, Alternaria solani, Trichoderma species, Microsporus, Alternaria solani, which is the disease is early blight in potato. Trichoderma species. Next, Microsporus, it is called ringworm disease. Ringworm disease in a human infection, itching, the plasma. So this is Microsporus. Clear? So this is entire information regarding Deuteromycetes. Now we have been discussed regarding Basidiomycetes as well as Deuteromycetes we completed. The entire fungal kingdom classes we have been discussed. In the next class we are going to discuss regarding virus, lichens and virions. Thank you.